In order to test my proposed hypothesis, multiple experimental techniques need to be used in order to prove it. The figure on this slide shows the overall layout of my experiment. First, I will take fibroblasts that have different ages from 0 to 5, 30 to 35, and 70 to 75 to indicate young, middle-aged, and old fibroblast cell lines. Then, at the first day and at 20 days, I want cells live stained for neuronal markers PSA and CAM, which specifically stains reprogram induced neurons but not the fibroblasts, and this is to get a visual representation of how many induced neurons were converted. Secondly, I want to do flow cytometry at these days in order to quantify the number of fibroblasts and induced neurons. And lastly, I want an immunocytochemical co-staining for neuronal markers beta-3 tubulin and NEUN. Then, on the other hand, I want to do RNA sequencing and neuronal transcriptome analysis to see if there is any transcriptomal differences at any of these days at 5, 10, 15, and 20 days. Alright, so for my experimental techniques, the groups that I will have are 3 donors in each groups. Group A will be fibroblasts from young individuals, Group B will be fibroblasts from middle-aged individuals, and C will be fibroblasts from elderly individuals that can be seen in the previous figure. So the first experimental technique that I want to use is following 3 weeks of conversion. Cells are live stained for the neuronal markers PSA and CAM, which stains reprogrammed induced neurons but not fibroblasts, and this will be the visual representation of the conversion. And so then I will want to use flow cytometry. And remember, flow cytometry is a technique used to detect and measure physical and chemical characteristics of a population of cells and particles. Then I want to do immunocytochemical for neuronal markers beta-3 tubulin and NEUN. And then in order to see changes induced on the transcriptional level, on the transcriptional level by different aged fibroblasts, I want to perform time series RNA sequencing from fibroblasts of each group. So what are my expected results? For the live staining, I expect to see more PSA and CAM positive cells in younger fibroblast conversions than those in older groups. For flow cytometry, quantitatively, the older cells will have less induced neuronal conversions than those of middle-aged or younger cells. So I expect to see a negative slope on the graph comparing young, middle, and old-aged fibroblasts. For chemical co-staining, younger groups, I believe, will have a higher expression of beta-3 tubulin and NEUN than those of older groups to show that there is a higher expression of induced neurons than those in the older group. And lastly, for the RNA sequencing, I expect to see some transcriptional level changes across different aged fibroblasts and possibly having to do with the methylation because methylation decreases as you age. So for the conclusions of this whole article, we have known that classic direct conversion protocols, which others have used to differentiate fibroblasts to mature human neurons, are highly inefficient and fail to successfully reprogram the majority of input cells due to cellular reprogramming. However, within this particular, it's using direct cell reprogramming because they use 2 different viral factors to co-infect fibroblasts to introduce NGN2 and ASCL1 proteins in them. But by creating a new vector construct that combines both NGN2 and ASCL1 transcription factors in the same vector, increases the performance of fibroblasts converting into induced neurons. The second conclusion is that by investigating the transcriptomal dynamics during reprogramming to identify the pathways that orchestrate induced neuronal conversion, scientists identified 4 small molecules that increase the conversion rate, which is a combination cocktail called ZPAC. Afterwards, scientists investigated the changes induced on the transcriptional level of the Z-Pak cocktail and found that the Z-Pak induced neurons were indeed more similar to brain expression patterns than previously thought, and scientists hypothesized that at the end it could have something to do with the maturity of the fibroblasts. However, further experimentation would need to be conducted in order to prove this. The last conclusion is that scientists confirmed that direct cell reprogramming in the Z-Pak cocktail could maintain epigenetic age, so they could look into age-related neurological disorders in future articles. And so lastly, the last slide I want to talk about is potential future work. As we've discussed, Z-Pak can cause a more thorough elimination of competing fibroblast programs on the transcriptional level than classic medium of NC alone. And specifically, scientists noted in this article that the inclusion of the ZM and PY small molecule could promote cytoskeleton reorganization and spare converting fibroblasts from mechanical stress-induced apoptosis. Another future work that could happen is that new phenotypes might be revealed from studying neurons in conditions that more thoroughly commit fibroblasts to neuronal fate specification, and in turn might lead to the discovery of more effective treatments for neurological disorders. And the last future work that scientists could do is using culture conditions that completely commit fibroblasts to neuronal lineage might result in an increased therapeutic potential of induced neurons for possible neuronal replacement. As I said before, there is work that uses induced neurons to replace damaged brain cells, but again, this would be future work that scientists could conclude. So thank you for listening to my presentation. That's all I have for you guys. I hope you guys enjoyed this presentation, and if you guys have any questions or comments or want to include any of your own proposed experiments, please leave comments in the discussion below, and I will try to answer back everybody's comments and questions as fast as I can. So thank you.